Hi everyone. So I'm Jeremy Kiska. I'm an associate professor of biology at Florida International University in Miami, and I'm really sorry for not being present in person today. Um, I'm currently in the field in the Indian Ocean on a tiny island, and out of precaution, we definitely thought it would be reasonable to record this presentation. So today I want to talk to you about some of the work that my colleagues and I did through a research program funded by the NOAA Restore Science Program, which was a 4 years project for which I got the chance to be one of the investigators. So, you heard a little bit about rice's whales, their habitat, their taxonomy, and right now I'm just going to talk to you about rice's whale food. Obviously, a really important component of the ecology of this endangered species. But before I start, I just wanted to thank my colleagues and co-authors. So this work was published earlier this year in the journal Scientific Report, and I would like to thank all of them listed here for their contribution. So before I get into the results, I just wanted to give you a little bit of background on why it is important to investigate the diet and prey selection in marine predators. The first reason is that it is critical to understand the role that animals plays within communities such as marine predators, and how important they can be in food webs. The second is that understanding the diet is really important because changes in the environment, prey distribution, or prey abundance and behavior can have significant consequences on predators. It sounds obvious, but that's really important for you to remember that, because not having the food that these animals require can have consequences on their body condition, their fitness, and their survival, and both at the individual and population levels. And investigating the diet of endangered species is great. It's important, but beyond the simple description of the diet of any animal, a major ecological problem is also to understand what drives prey selection. Why does the observed prey or array of prey species compose the diet of a given predator? And answering this question is really important to predict the potential effects of human activities on resource availability, for example, due to fishing or climate change. And the optimal foraging theory is a classical paradigm to explain and predict the diet of a forager. And the general idea is that animals are adapted to use resources as efficiently as possible. And there are a few major fundamental predictions, but there's two that I wanted to mention here. Is that predators and foragers in general should prefer resources that yield more energy compared to the cost. That's kind of obvious, right? And as abundance of high quality prey, those that are more profitable, increase in the environment, lower quality prey, those that has less profitability, should be dropped out from the diet, and therefore those animals, predators, like rice's whales, should become more specialized. Cetaceans are really important predators in marine ecosystems. They can really have significant impact on community on communities and ecosystem dynamics through multiple processes. And the role of predators is pretty much where they are in the food web, right? And cetaceans exhibit a wide range of roles. They feed on many different prey, and they employ a variety of different tactics. But interestingly, since those animals are so difficult to study, very little is known about active prey choice. Why do they choose their prey? Well, there are many studies describing the diet of these species, of these predators, but very few, it's really a handful, that actually explore the drivers of prey selection. And obviously it's difficult to address this question. Why? Because it's very difficult to quantify the diet and how it varies, spatially and temporally. But it's also so difficult to understand the prey, and that's a critical component of this question. The question of prey selection. You need to understand where the prey are, and also what's the intrinsic characteristics of those preys, including their quality and their energy content. I'm going to go very briefly here because you've heard of it. Rice whales, an endemic to the Gulf of Mexico, and we know very little about these animals. And one thing that we do not know at all, at least before this work was done, was their feeding ecology, their diet. We knew a little bit about their diving behavior, but based on one individual, which was something that was published a few years back. Now we had another individual confirming quite similar patterns in terms of diving behavior, but their diet really was not known. And how do you study the diet of cetaceans? So if you cannot observe constantly, continuously these animals, you have to use other methods. And the most widespread, I would say, and the most commonly used, especially in the past, not necessarily today, to study the diet of cetaceans, is the examination of stomach content. And usually it's limited to animals that are stranded or captured, either incidentally or on purpose, since that's the case in some places around the globe. And it's definitely the most comprehensive method because it allows you to really identify prey at the species level. Of course, it has some issues, but really that's the most exhaustive way, most comprehensive way to quantify the diet of marine predators. The problem is for rice whales, we have very, very few strandings, and the few strandings that we had were of individuals that were in poor body condition or already sick, so there were no content. So it's a little bit challenging, but you have alternatives today. And stable isotope analysis is an indirect method that is really commonly used to study the diet of marine predators, especially cetaceans. So basically, and very quickly, stable carbon and nitrogen isotope analysis give us some insight into the trophic relationships of organisms within communities. And carbon isotope values are indicative of where the animals are feeding, where in the wallow column. Is it close to the bottom? Is it close to the surface? We can actually figure that out using carbon 13 values. And nitrogen 15 values, isotope values, are indicative of the trophic position, where in the food web the species that we study actually feeds. And while stable isotope analysis is a great method to study the trophic interactions of cetaceans, and it can be done from a tiny piece of sample, such as a skin biopsy that can be remotely collected with a crossbow. The problem is that it lacks quantitative prediction, But there's been what we call mass balance models that have been developed to determine the relative contribution of prey sources based on stable isotope data from the predator and from potential prey. So multiple potential species that you sample, and using stable isotope, you can actually reconstruct using what we call the trophic enrichment factor. So if you know what's the isotopic difference between the prey and the predator, you can then produce those, which are proportions of in the diet of a predator of multiple prey. So here, this is typically what we did with rice as well. So in a form of probability expressed here in percentages, you have estimates of the source contributions of rice as well. So I'm gonna talk to you about that now. So what do? We collected 10 biopsy samples from 2010 to 2019, and we collected potential prey using troll surveys that we did in July 2019. So those troll surveys were done both to collect samples, but most importantly to understand the abundance and species composition of fish and other potential prey within the rice as well habitat. So we used stable isotope mixing models that I briefly described before. We looked at prey availability using troll survey, and the samples were again used for stable isotopes, but also to look at the proximate composition of each species. Basically, we looked at the energy content and therefore prey quality in the diet of rice as well. And we used a few indices, the Chesson index or the Pianka index, actually to look at active selection, and also if there's a match between what's available in the habitat and what is actually consumed by the whale. So, you remember the graph that I showed you before. Well, this is basically what we got here. And what's really striking is that rice as whales consume at nearly 70% a demersal schooling fish called Ariomma bondi. Other species that we sampled and that were available in the habitat had a relatively minor contribution. And so, that was the most significant result that we've got here. So we have rice as whales that have a fairly clear pattern in terms of their diet. During 21 troll surveys that we carried out in the summer of 2019, we recorded 26 species of fish, cephalopods, so that's squids primarily, and octopus, and invertebrate. And as you can see on this graph, really like 4 species dominated. You'll have to trust me on that one. It's probably a little difficult to read all the graphs, but really we had 4 species that dominated and that actually dictated our choice of prey, of potential prey for stable isotope analysis. And what's interesting is that Ariomma bondi was abundant, but it was not necessarily the most abundant species. Morellicus white's money was actually numerically speaking the most abundant species. Ariomma bondi and Morellicus were probably equally abundant in terms of biomass, and then other species like Diaphus dumerli and Dorythotis, which is a squid, were also fairly abundant. Not really Diaphus, which is a small lanternfish, in biomass, but really we had like 3 prey in terms of biomass that were quite abundant. The energy density calculated for rice as well prey revealed some major differences, and here's the take-home message. When you look at certain indicators of prey quality, like the content, the lipid content, and the protein content, Ariomma Ariomma bondi, excuse me, were definitely the highest quality prey. And what's really interesting from the different indices that we calculated is that, you know, there's a relatively low degree of overlap between what is available in the ecosystem and what is actually consumed by rice as well, especially based on the Piankett index. So it really suggests that prey abundance is not necessarily the main or the primary driver of prey selection. And the Chesson index that we also calculated revealed that only 3 out of the 4 species identified as prey species were actively selected. And we also think that some species like Morellicus weinsmanni here on the left, on the right-hand side of the graph, was maybe a byproduct of other species consumption, since it's so widely distributed in the water column. But their contribution was relatively small. If you're some of the diving behavior information that I was referring to a little earlier, because rice whales forage near the bottom and undertake relatively costly foraging dives, sometimes exceeding 250 meters, where Ariomma bondi actually occurs. So, this work is important because I think we have our, first information on what these animals are actually doing and eating. And in terms of conservation management, this is important. If there is any change in the behavior and the distribution, the vertical movement of Ariomma bondi in relation to climate change, for example, that could have potentially dramatic consequences on rice as well. All right, well, that's the end. Thank you so much for your attention. That's the acknowledgement. Also wanted to share a graphical abstract that we put together with Anaïs Remily, and I'm happy to share it if you need. Thank you so much for your attention.